In this video, we will discuss about some of the application of RBPJ knockout activated T cells in the in vivo, that is in the mice model, and its impact on the FOXP3 expression and also in the probability of its survival. So in this case, they took the NSG mice, and for the control they directly inject this PBMC, and in other case they inject they aggregate this T cells and inject them as it is, and in one case they knock down this RBPJ and inject it, and in other case they activated these T cells inside this and transduce this or inject them into these mice models. And what are the results that they are able to find out is that when they use direct the PBMC, the survival rates are minimum, and when they use activated T cells with PBMC, they do not see any increase in the survival rate. But when they use the normal T cells with PBMC, they are able to see some increase in the survival rate. And when they use the activated T cells with the RBPJ knockdown, they can see the same survival rate as with the normal T cells. This indicates that this RBPJ has a role towards this suppressions of the FOXP3, which might be causing in the increased survival rate in the case of RBPJ mice and the normal T cells. That can be seen here. In the activated T cells, in the normal case, we do not see any increase in the survival rate because RBPJ is present, and that is suppressing the FOXP3 expressions that can help in the increase in the survival rate. And when they check the impact on the weight of the mice model in the 4 different conditions, what they are able to see in the case of PBMC, they can see the more body weight in the mice with PBMC percentage is decreasing with times at shortest period of time, and with then when they use normal T cells, they see the more increased or less decrease in the body weight with time. And when they use the activated T cells as a control, in that case also they are able to see some body weight decrease at short duration of time, like around 20 days, 15 days, and the body weight continues to go at the constant rate is low. But when they use the activated T cells with RBPJ knockdown, they are able to see most of this body weights are continue to go even after 30 days. Thereafter, they wanted to see, they also check the FOXP3 expressions, whether it is also get affected or not. So when they check it in the flow cytometer before transfer, they can see that in the case of this activated T cells as a control, the FOXP3 expressions around 89.6%, and in the case where RBPJ has been knocked down, they can see FOXP3 expressions is around 91.5%. And post 5 days after transfer, they are able to see that FOXP3 expressions goes down in both the cases compared to the first before transfer case. And when they checked, they quantified these things in the quantified this flow cytometry results of the penalty, they can see that in the RBPJ knockout, the FOXP3 expressions are significantly higher compared to the control case. And for the 5-day post transfers also, although the difference decreases, but the increase is still statistically significant in the RBPJ knockout compared to the control case. So this indicates that RBPJ has a direct role towards the FOXP3 suppressions even in the in vivo conditions that is checked in the NSG mice model. This validates their previous in vitro results that they have seen about the relationship between FOXP3 act as a negative regulator for RBPJ act as a negative regulator for FOXP3 expressions. And this can be used as a one of the therapeutic targets in future to increase the efficiency of this to increase the efficiencies of these T cells. So I hope you understood the results that are presented in this figure. Thank you for your time.